Hello, I am the esteemed Dr. Panda. I am delighted to be sitting here today with the Channel Teddy News crew. As I understand it, I am here to discuss the affairs of junk DNA. In our recent discoveries, we have addressed such breakthroughs as identifying regions that may regulate certain protein intake and other regions that may promote RNA transcription within the genome element.
Amazing, Dr. Panda. Can you tell us why you are all black and white?
You see, little lady, it all has to do with genes and my DNA.
What does all that mean exactly?
It's called science. The cells in my body are made out of proteins. These proteins carry a lot of information inside their nucleus to help regulate my bodily functions. Some functions were chosen before I was born, such the color of my eyes and the shade of my fur.
Yes, doctor, we are all aware of the discoveries made by Sir Watson and Crick. An amino structure designed to duplicate itself and carry hereditary information. Tell us more about the part of the DNA that does not do anything.
Junk DNA is turning out to actually not be junk at all. These regions hold very specific data to regulate and construct numerous, unbeknownst, active and ongoing organic cycles. The fine gentlemen done at the ENCODE project headquarters are looking into exactly which sequences do exactly what actions in exactly which positions.
I'm going to interrupt you right there and ask this question. Why do I care about all your blah blah blah?
The answer, once again miss, is science. The knowledge we can glean from such little building blocks could help us build and engineer biotechnology and medicine. Knowledge is power.
So, please pray tell us, I know the world is dying to know, what piece of technology is used most commonly in the lab?
In order to read the many lines of deoxyribonucleic acid, a sequencing machine is used. It is a big tool with lights and mirrors that tells us what the DNA says. The machines get faster and cheaper every year.
Oh, you do not say. Please do go on.
In a decade or so, who knows where the exponentials of technology may lead us. I like to speculate that there will very specific breeding in the future. What if my cubs were white and black, rather than black and white? That would be terrible. New technology will be able to let us go inside and alter, possibly even create, an embryo and pick and choose the traits we want our children to have. Maybe we would even like to clone them.
Go ahead and talk to the children, the future workers of the 1%.
Many technological institutions offer courses to become lab technicians and phlebotomists. ITT will get you that degree, children.
This just in, advanced biotechnologies such as selective mutating and cloning could lead to harmful bioweapons and be very hazardous to the people. The government is rendering sanctions to pull any research off the shelves.
But look at the very positive side of things. Such technologies will help improve the day-to-day life for all of the people. Someday we may have the ability to create unicorns and flying monkeys or flying monkey unicorns. Although, the research may be impeded at first due to the lack of social agreement with the ethics behind biotechnologies and DNA engineering. A more complete understanding of the junk DNA brings us closer to the origin of all species and might tell us where we are headed on the pale blue dot of ours. If people begin to realize the differences in the human DNA from one race to another is so minute that it doesn't even matter. People might begin to realize, with the completion of mapping the DNA, that we as a species come from one very strange creature and we are all bears on the inside. Our greatest achievement with unlocking the secrets of life would be the acceptance of love.
Looks like this concludes our enthralling look into epigenetics and junk DNA. We learned that genes control who you are and may point to who you will be. Join us next week when we investigate more into the exciting life raccoons. Are they as cool as they think we think they are? Riveting.